Dr. Stefan Halbherr from the Bern of this Switzerland. Okay, his talk will be about the glycan-targeted liposomes.
Hello, everybody. Good afternoon. Thanks for staying that long already. I'm happy to present to you our latest proceedings where we have been focusing on controlling biodistributions with liposomes, as many probably have done. And I can tell you now already, one of our special features is that we attach sugar chains on top of the lipid layer, which has some pretty interesting effects. So to give you a brief overview, I want to introduce the nature of glycans a little to you, why they are there, what they do, and where they actually are in biology, where they are located macroscopically and microscopically, what kind of liposomes can be derived from using glycans, and what kind of architectures are possible. A couple of examples, what we have tried in vivo, whether or not these surface modifications enable cancer targeting or also different patterns of biodistribution. And last, we investigated, of course, the therapeutic potential behind this. So besides glycans as a surface modification, liposomes are influenced by a myriad of other factors, such as the lipid mix itself. You can imagine charge plays a very crucial role. The process, the way particles are assembled, is a very important factor, which is also determining the size, the lamellarity, the stability, and so on. There are 20 different methods at least available. We haven't really tried all of the possible combinations, of course, otherwise we would have done this for more than a human life. But just to mention a little of the theoretic background, of course, drug loading takes an impact, and at least when you have all these factors in a constant, you can start to address the surface modification specifically and see what's the potential behind it. Just a couple of remarks to glycans. As you are all probably familiar with, they are known basically that they are there, but the functionalities are rather, I would say, not as well understood as actually DNA and protein are related and understood already. And however, they are a lot like DNA and protein in a way that they are derived from 9 different building blocks. DNA is derived from 4 blocks, and proteins are derived from a couple of amino acids. But thereof, there has been estimated that around 7,000 different glycan structures at least exist in humans. And they all have different affinities to different lectins, which is a very interesting feature because unlike antibodies, which are usually only hitting a very specific target that usually, I think in nature, are rather virus-like proteins actually that are neutralized by antibodies very effectively. Glycans have a more promiscuous binding behavior. They have low and higher affinities, but not in the same range actually as antibodies. So one of the very good features of using lectins is that they are very high expressed and surface accessible, making them pretty interesting targets. Using glycans on top of the liposome has an advantage in a way of hydrophilizing the sort of hydrophobic lipid membrane, and making liposomes very hydrophilic is good in a way that it conjugates a lot of water around the liposome and giving it less chance for aggregation and more stability, and of course targeting effects. Glycans have been shown to play very important roles in the host-pathogen interactions, which is the pathogen is actually the nature's best way of entering into a cell. And if you have your mission that you want to deliver a drug into a cell, which we do at least for chemotherapy, maybe you can get some inspiration from nature and think like if you are a virus, how do you get into a cell? Or if you're a bacteria, how do you get into a cell? And apparently viruses as well as bacteria, they like to use glycan-lectin interactions and really effectively get into a cell and deliver whatever they should not deliver actually if you're a human. But yeah. Next, I just wanted to outline quickly, in 2013, the Nobel Prize of Medicine went to intracellular vesicle trafficking. And interestingly, the Golgi apparatus is putting very specifically glycan building blocks on top of each other while the vesicles traffic through from the cis Golgi to the trans Golgi. And this has an influence on the localization of the particle itself inside of the cell. And what we have been asking ourselves is what actually if also extracellular vesicle trafficking is at least to some part guided by glycans could be. There were already a couple of papers available, but not that many. So as you can see here, this is our experimental approach. On the left side, we have started with a base vesicle, and we kept this very constant. And there are a couple of different technologies available by which you can modify the surface. We used amine reactive chemistry with linkers. And by such, we attached serum albumin, we attached polyethylene glycol, or we attached also just very short linkers. And the type 1 vesicle I will not further discuss in this session because it takes too much time, but actually with PEG and HSA we have done quite some work. So let me show you a couple of biodistributions we have been measuring. For that, we used liposome formulations carrying 10 micrograms per milliliter of near-infrared dye. They did not have any payload to simplify things, and we injected 250 microliters per mouse, and the excitation emission was in the near-infrared range, of course. So a untargeted particle carrying only serum albumin actually within 4 hours is found to a large part in our hands inside of the liver and not so much into the tumor, which is indicated by the yellow arrow and the left flank of the mouse. After 24 hours, this picture doesn't change, but if you add sugar, which we call for now just SC sugar chain 2, SC2, glycan, the biodistribution sort of seems to change, and there's accumulation visible inside of the tumor burden. And the same is also true if you change the glycan to a glycan we call A6. If you want to know what it is, maybe rather come later to me and we'll have a discussion. If we put those all together on the side, you can see that, some targeting and some tumor accumulation can be derived by using glycans. I can tell you now already it's not monosaccharides. Monosaccharides you can forget, they will, or unless you want to target the liver, you can very effectively. All of the monosaccharides we tried directly went into the liver. I forgot to tell you that. So next we did type 2 vesicles where you used polyethylene glycol. This is a negative control which has an interesting biodistribution pattern where at least some accumulation is found in tumors. But there is also a quite diffuse pattern where you see the lung on top of the liver lighting up a little, and also quite a diffuse pattern inside of the abdomen, which is probably also what we know from literature so far about the human evidence of these particles. Now, very interesting, when you add a sugar chain on top of this, the distribution narrows in quite nicely to the tumor as well as the liver again. Interesting point is when you change, this is the sugar chain 4, I would like to tell you this is a C-allele Lewis chain, the natural ligand binding to A-selectin, gives a very significantly different pattern of biodistribution, and I cannot tell you what we could use this for so far. I think it's multi-organ targeting, and I don't know, depends what you want to do. Maybe you have a disease where this is useful, I don't know. But so far, we just use this as a proof of concept that sugars have a great impact on the biodistribution, and that sooner or later there will be interesting things derived thereof. This is the C-allele Lewis A, which is very similar to C-allele Lewis X, but just a minor, very small difference, giving a quite similar picture of distribution. This is one of our last designs of vesicles where we just added the pegylation by a soluble linkage. So it wasn't incorporated into the vesicle by normal post-insertion technique or pre-insertion technique. We just linked it on top. So it's just outside of the vesicle, which gave again the same biodistribution as we also saw when it's inside, but it's just, yeah, we were trying. When we compare this, all different particles in parallel, this is what you get. Oh, I have to hurry up. Therapeutically, these vesicles showed, I would say, beneficial effects, or at least potentially beneficial effects when we treated ovarian cancer xenografted mice carrying A2780 human ovarian tumors. Mice were treated twice weekly after the tumors were grown for like 8 days. So there was an initial starting burden, and mice were treated for 3 weeks consecutively, and there was 1 week of relapse where tumors were allowed to regrow. And you can see free doxorubicin doesn't really impact on tumors that have relapsed anymore, whilst nanoformulations do. And there might be a potential benefit derived from this TLD1 formulation. Just as a side remark, the TLD formulation we manufactured amongst the surface modifications, it is slightly smaller in size than commercially extruded vesicles. It's around 60 nanometers, whereas the extruded ones are rather around 100, and this might have an impact on what we found as renal clearance, actually, which is quite elevated for this particle. Second, we went for a last formulation, which we call TLD2, and it showed a very nice tumor accumulation in this near-infrared imaging. And what we saw in a therapeutic study actually was quite surprising, that the toxicity of this particle was actually elevated. And you see on the, I showed this in the green curve on the bottom, you see that the body weight is slightly it's more dropping than for the other compounds, even though we only treated it 3 out of 7 times of treatment. So I think this is the first time where we were able to demonstrate actually dose reduction, not a dose elevation, which is usually done, that you just administrate more. And we believe that this is actually what should be done. You should be, at the end, if you really hit the target, you should be dosing less. And I've worked a lot with doxorubicin. I think it might be desirable to really have less of doxorubicin inside of your body than having more of it. I'm really afraid of having a long-term toxicity effect when you just give more of this substance, because we all know the effects, and basically it cannot be like eating a carrot or so. So to sum up, biodistributions are quite significantly influenced by using PEG or HSA. Surface modifications by sugars allow for very different biodistribution patterns, and we were able to measure potentially beneficial effects compared to free drug or commercial liposomes, as well as we were demonstrating first evidence of a successful dose reduction by more than 50% without evident loss of efficacy. However, there was toxicity which we will address in a second in following up experiments. Well, we do further, of course, reach out our research in oncology, and we look at nanonutraceuticals, which might be an interesting way of using resveratrol or something alike inside of a liposome. And we aim at clinical translation sooner or later. I want to thank the InnoMedica team, especially the scientists, as well as financial administration and the company board, and last, of course, our investors. And this project has been realized in collaboration with the Mario Negri Institute in Milan, as well as the University in Bern. And if you have any idea of a liposome you would like to try but cannot really make, or you think you want to give the glycans yourself a spin, give me an email. I will happily send you a couple of milliliters or liters, depends what you want. We have a GMP facility, and we can do quite some scaling. And if you have some questions, I'd be happy to answer them.
Okay, thank you. And please.
So is the glycan actually binding to a receptor on the cancer and then inducing endocytosis? I didn't catch this part.
Yeah, this is very difficult to actually tell. Since cancers, we have never really done in vitro studies because we were really interested in this in vivo situation where you have to take into account the clearance of the liver, the accumulation of the tumors. So we never really ex vivo laid down some cancer cells, and well, we did, but it wasn't really interesting and allowing for any conclusions. But so we cannot really tell what is the target of the glycan. Since when you know the glycan structure, you can derive maybe the one or other lectin. You think, maybe this should bind to this. But actually, since the whole in vivo situation is so much different than in vitro, I thought, even if I wanted to, I do not know any in vitro experiment other than just checking the stability of the particle, of course, that would allow for targeting.
Yeah, please, continue.
Can you, for example, use free glycan to block the uptake, for example? I mean, is it a saturated? The key question, if it's a receptor, then you can identify which cancer maybe can be treated, right?
Good suggestion. No, we never tried.
Yeah,
a couple of glycans are very expensive. The other ones are rather cheap. So, well, with the cheap ones, we could try. Yes.
So, in line with his question, you made the xenograft in the liver.
No. The xenograft was on the left flank.
So, it is anyway the human, the ovarian cancer.
Yeah.
Was implanted subcutaneously, but why did you just put near liver? So you did not a priori you did not expect the liver would have taken up your the glycan label like.
Well, this experiment, we only manufacture the liposomes at InnoMedica.
Yes, okay.
Other research groups do the in vivo trials, actually. And I don't know why they put the tumor always there.
So confusing. Yeah, so. And also the next question is just according to the Wikipedia, silyl Lewis A is tetrasaccharide, which is known to be the target of the CA19-9. So it means that it is just a kind of the same antigen in the colorectal cancer, mostly. So, but you have used the ovarian cancer model. So confusing as a physician. I'm a physician. So
yeah, I'm confused too. But what the sense behind this is that we have been using doxorubicin as a model drug. And we want to compare against free drug and commercially liposomal products that are quality certified and a really stringent way of controlling untargeted versus targeting. Second, in human clinics, doxorubicin is used for ovarian cancer.
Yeah.
So makes a little sense at least.
Okay, one request for short clarification and one question. The clarification, are you loading your drug in the same way like the commercial doxil, and whether it is leaving the liposomes at the same slow pace, right, like the commercial calyx and doxil? This is clarification. And the question is, so sugars usually are analytical nightmare. So there are problems with stability, with characterization, with other analytical issues. Can you please refer to this shortly?
Yes, absolutely. So I think the first point I will be able to tell you next year, because we had a sort of bottom-down approach, top-down approach, sorry, where we were first actually going in vivo after initial assessment, and then when we kept animal numbers very small because of ethical considerations. But at the same point, we thought we have to go in vivo, because otherwise we will never know actually what is enabling tumor uptake or whatever impact on biodistribution. So I will tell you next year probably. And the second thing is, it was a very difficult step for the startup company to master Glycan analytics. We are still only for sugars that have sialic acid actually are detectable and quantifiable. However, it comes with the problem that you only detect the sialic acid and not the rest of the glycan. So we will hopefully soon buy a mass spectrometer, and our analytical guy will need to clone himself twice or so. Then we can go further with this. But so far we know that the sugars are bound on top of the vesicles, and this is what we have been measuring.
Okay, thank you. And thank you.
Thank you.
Yeah. Before closing the session, if you have any question you forgot to ask, just like myself, I have a question to Dr. Lino. He just told us about the kind of Trojan horse, as I named it, the type of nanoparticle laden, the leukemic cell approach. And you, in your slide, I just forgot to. Something, it's exosome. So you are suspecting that the differentiating cells, because of the nanoparticle, excited by the laser, are emitting exosomes carrying some messenger to just kind of adjacent leukemic cells to differentiate to. Oh, that was a fantastic, if if it is proved, solidly. Okay, does anybody have any question? If Professor Wolfgang Parag would have been here, I was just trying to ask him, why? Because we have 20 or more minutes, ask him about his original contents. Actually, he just was trying to throw the bomb. Yeah, so why? Because when you are measuring some fluorescence labeled or radioisotope labeled or other types of the marker or reporter labeled nanoparticles and whereabouts of the in vivo distribution, then you need to check if the label is detached, and you're just observing the fluorescence marker or the radioisotope in the capsule. So that might just kind of might have enlightened us more. So just like the last presenter's presentation, we need to come again here next year to listen to his the continuing talk. And because I was trying to, is there anybody to say something? I was trying to save the time for the presenters, so I did not introduce myself. I am Dong-Sul Lee. Actually, I'm a nuclear medicine physician, majored in radioisotope things, and I'm going to present a radio nanomedicine tomorrow, the same time here. And also I am going to, next year we are going to have the summer school in Barcelona, as you remember. I wonder if you remember or not. The Professor Hunziker, president of the ISNM, told us during introduction Next year, we are going to have a summer school for the junior people. And this year, first summer school is going to be coordinated by myself. Thank you. Thank you for your participation.
To make a remark from the long list of cooperators that I made, I forgot the most important, which is close to here, from Lausanne, from the EPFL. We are also cooperating with them now.
Okay,
Sorry.
thank you. Thank you. Yeah, I'd like to close.